Welcome to biotanime.
HSP-100. HSP-100 A CLP is a class of molecular chaperones that have the ability to solubilize almost any protein that becomes aggregated after severe stress. Structure and classification. CLP or HSP-100 proteins assemble into ring structures. It has an N-terminal domain followed by a nucleotide binding domain or NBD1. A middle domain and a second nucleotide binding domain or NBD2. HSP-100 class of chaperones belongs to the AAA+ superfamily of Itpaces. The family is defined by the presence of a basic core of approximately 200 to 250 amino acids that comprise alpha helical domain and a Walker-type nucleotide binding domain. They have the ability to remodel the protein substrate in an ATP-dependent manner. HSP-100 chaperones are categorized into two classes. 1. Class 1 proteins with two AAA+ modules. For example, HSP-104, bacterial CLPB and their distant relatives CLPA, CLPC. 2. Class 2 chaperones. Example, CLPX and HSLU. Machinery for protein disaggregation. The nucleotide binding domains of HSP-104 and CLPB contain the characteristic Walker A and B motifs and sensor 1 and 2 motifs. Both the nucleotide binding domains are capable of binding and hydrolyzing up, and the ATP binding to the nucleotide binding domain stabilizes both the oligomeric state and interactions with the substrate. Hydrolysis of ATP provides energy for protein remodeling. The presence of M domain is unique for HSP-104 and CLPB proteins and distinguish it from other class 1 proteins. It consists of two anti-parallel coils resembling a two-bladed propeller and is inserted in the ATP binding domain, which is essential for protein remodeling activity of the proteins. Most of the disaggregation function by CLPSP-104 is carried out in collaboration with the HSP-70T and AK chaperone system. AAA+ proteases or ATP-powered protein degradation compartments. HSP-100 form an important subfamily of AAA+ machines that function in ATP-dependent protein degradation in cells ranging from bacteria to humans. Examples of ATP-dependent proteases include CLPXP, CLPAP, 42-CLPCP, HSLUV, LON, FTSH, PAN20S, and the 26S proteasome. CLPAP was the first AAA+ protease that was identified. CLPAP consisted of CLPA that functioned as an Itpase and a separate peptidase CLPP. Degradation of protein substrates required a combinatorial action of CLPA, CLPP, and ATP hydrolysis. CLPX is AAA+ protease that carried out ATP-dependent proteolysis of substrates such as phage replication protein lambda O. CLPXP is composed of two distinct proteins, AAA+ Itpase called CLPX, and a peptidase called CLPP. CLPX recognizes unstructured peptide sequences in protein substrates and unfolds stable tertiary structure in the protein. The unfolded polypeptide chain is then spooled and translocated into a sequestered proteolytic compartment in CLPP for degradation into small peptide fragments.
Thank you. Please like, subscribe and share. See you again in a new topic.